July 25th, 1978, Louise Joy Brown, the world's first baby produced via in vitro fertilization, is born to parents Leslie and Peter Brown. The healthy baby was delivered by cesarean section and weighed in at 5 pounds, 12 ounces. Before this, Leslie Brown had suffered years of infertility due to blocked fallopian tubes. In vitro is Latin for within the glass. When something is performed in vitro, it happens outside of a living organism. In November 1977, Leslie Brown underwent the then experimental IVF procedure. A mature egg was removed from one of her ovaries and combined in a laboratory dish with her husband's sperm to form an embryo. The embryo then was implanted into her uterus a few days later. So basically, they take the egg out, they put it into a glass container, and then put the sperm in and fertilize it together within the glass container, grow it to an embryo, and then throw that sucker back in to the uterus. IVF doctors, British gynecologist Patrick Steptoe and scientist Robert Edwards had begun their pioneering collaboration a decade earlier. Once the media learned of the pregnancy, the Browns faced intense public scrutiny. Louise's birth made headlines around the world and raised various legal and ethical questions. The Browns had a second daughter, Natalie, several years later, also through IVF. In May 1999, Natalie became the first IVF baby to give birth to a child of her own. The child's conception was natural, easing some concerns that female IVF babies would be unable to get pregnant naturally. In December 2006, Louise Brown, the original test tube baby, gave birth to a boy, Cameron John Mullender, who also was conceived naturally. In 1996, a female sheep gave birth to the now famous Dolly, a Finn Dorset lamb. According to History.com, Dolly the sheep was the first mammal to have been successfully cloned from an adult cell. Originally codenamed 6LL3, the cloned lamb was named after singer and actress Dolly Parton. The name was reportedly suggested by one of the stockmen who assisted with her birth after he learned that the animal was cloned from a mammary cell. The cells had been taken from the udder of a 6-year-old ewe and cultured in a lab using microscopic needles, in a method first used in human fertility treatments in the 1970s. After producing a number of normal eggs, scientists implanted them into surrogate ewes. 148 days later, one of them gave birth to Dolly. Dolly's birth was announced publicly in February 1997 to a storm of controversy. On one hand, supporters argued that cloning technology had led to crucial advances in medicine, citing the production of genetically modified animals to be organ donors for humans as well as therapeutic cloning, or the process of cloning embryos in order to collect stem cells for use in the development of treatments for degenerative diseases. Dolly was mated to a male sheep named David and eventually gave birth to 4 lambs named Bonnie, twins Sally and Rosie, and triplets Lucy, Darcy, and Cotton. Interesting names, a couple biblical names in there. In January 2002, she was found to have arthritis in her hind legs, a diagnosis that raised questions about genetic abnormalities that may have been caused in the cloning process. After suffering from a progressive lung disease, Dolly was put down on February 14, 2003, at the age of 6. Some scientists have looked at human cloning as a possible way to preserve endangered species, and then others view it as unsafe and unethical, especially when applied to humans. Officially, Dolly was created using the technology of somatic cell nuclear transfer, which we've talked about before, where the cell nucleus from an adult cell is transferred into an unfertilized developing egg cell that had its cell nucleus removed. It should be noted that there was a sheep cloned before, but it was using embryonic cells instead of adult cells. Whatever made Dolly, the big one, I'm not sure. 2 other sheep Megan and Morag had also been cloned from embryonic cells grown in the lab at the Roslin Institute in 1995, and 6 other sheep cloned from embryonic and foetal cells were born at Roslin at the same time as Dolly. What made Dolly so special was that she had been made from an adult cell, which no one at the time thought was possible. Them using an adult cell meant that they could take any adult species, whether it be a superstar celebrity, athlete, or an old extinct woolly mammoth. The Journal of Cell Biology stated on Gurdon's frogs, in the case of Dolly, these experiments, together with the more recent cloning of Dolly the sheep, revealed the potential of nuclear transfer for cell replacement therapy. The potential would be even greater if these cells could be reprogrammed directly without the need to transfer nuclei into eggs, a scant resource in humans. To this end, Gurdon has spent much of his career deciphering the molecules and mechanisms that the egg uses to rejuvenate nuclei. And that's a great word for it, rejuvenate instead of reprogram. In 1997, the National Bioethics Advisory Commission concluded that attempting to clone a human being was at this time morally unacceptable. They recommended continuing President Clinton's ban on the use of federal funds to support the cloning of humans, and called for federal legislation prohibiting anyone from cloning a child during the next 3 to 5 years. Scientist woo-suk first caught media attention in South Korea when he announced he successfully created a cloned dairy cow, Yeon-grong-I, in February 1999. His alleged success was touted as the 5th instance in the world in cow cloning, with a notable caveat. Hwang failed to provide scientifically verifiable data for the research, given only media sessions and photo ops. Hwang would be a mysterious character in the world of cloning. He would go on to, be a controversial figure where he would, do photo ops, but no scientific data turned in. So you never know if he's really hiding the real stuff or if he's just a fake guy from the start. Hwang's several claims were well received by South Korea media and public, who were attracted to his claims of economic prospect, and so on. Hwang's main research was in creating genetically modified livestock, including cows and pigs, and he claimed to create a BSE-resistant cow, which is mad cow disease, and he also stated his intention to clone a Siberian tiger. In February 2004, Hwang and his team announced that they had successfully created an embryonic stem cell with the somatic cell nuclear transfer method we'd been talking about, and they published their paper to the March 12th issue of Science. Although Hwang had already established himself as an expert in animal cloning and secured his celebrity status in South Korea in the late 90s, his alleged sudden success came as a surprise because this was the first reported success in human somatic cell cloning. Until Hwang's claim, it was generally agreed that creating a human stem cell by cloning was next to impossible due to the complexity of primates. Hwang explained that his team used 242 eggs to create a single cell line. Hwang would announce an even greater achievement in May 2005 when he said that they created 11 human embryonic stem cells using 185 eggs. His work, published in the June 17th issue of Science, was instantly hailed as a breakthrough in biotechnology because the cells were allegedly created with somatic cells from patients of different age and gender, while the stem cell of 2004 was created with eggs and somatic cells from a single female donor. Hwang was opening the doors of possibility to cloning. This also meant that every patient could receive custom-made treatment with no immune reactions. Time magazine would name Hwang one of its People Who Mattered 2004, stating Hwang has already proved that human cloning is no longer science fiction, but is a fact of life. Following on the earlier success, on August 3, 2005, Hwang announced that his team of researchers had become the first team to successfully clone a dog, which has been independently verified through genetic testing. The dog An Afghan hound was named Snoopy. Even though his career was seen as suspicious and shrouded with mystery and controversy, he was able to keep his biotech company, and he teamed up with Boyle Life Group to hopefully make in his factory produce 1 million cattle embryos per year to meet the increasing demand for quality beef in China. So they're just eating nothing but cloned beef. I should mention that his company Soam Biotech is very similar to Repet's in the movie The Sixth Day. So if you've seen that movie, he basically runs Repet's in a way. This is actually from the website itself. You see how cloning is so easy? The whole process involving, let's say it was your dog is the top right box, somatic cell from donor. The rest is everything else that they do. So it's so simple.
The cloning, say no to Repet.
We can clone your four-legged loved one in just a few short hours.